signal to the nervous system. The motor neuron relays an action potential to its connection with the muscle cell, which in a bundle is called a fascicle. Calcium channels on the axon open, and the calcium forces synaptic vesicles to the end of the synaptic knob, where they release acetylcholine into the synaptic cleft via exocytosis. Choline binds to acetylcholine receptors, which are located on the motor end plate. The chemically gated ion channel is open when its meeting happens, allowing a flow of sodium ions into the muscle cell. After the acetylcholine opens the sodium channels to depolarize at the motor end plate, acetylcholine esterase breaks the acetylcholine down into acetic acid and choline, which absorbs back into the synaptic knob and eventually binds back into acetylcholine inside the synaptic vesicles to restart the process. Back to the muscle contraction, the depolarization of the motor end plate activates the sarcolemma and stimulates another action potential. Moving through the transverse tubules, the action potential gets to the myofibrils inside the myofiber. Encasing the myofibrils are sacs called the sarcoplasmic reticulum. This arrival of action potential causes the excretion of calcium ions kept inside the terminal cisternae. It is here that calsequestrin sequesters calcium ions. Potential has reached the muscle fibers. These fibers are arranged in units called sarcomeres, and they contain both thick and thin filaments. The Z-disc is the length of the sarcomere where thin filaments attach. The A-band is dark with thick filaments and some thin filaments. The I-band is the light band, which is composed of thin filaments, and the H-band is the light section of the A-band. Thick filaments are composed of myosin, and the thin filaments are composed of actin, troponin, and tropomyosin. When calcium from the terminal cisternae is released, it attaches to the troponin, causing the troponin to pull on tropomyosin and expose the active sites. These are irresistible to the myosin head, causing them to reach out and attach with the adenosine diphosphate and phosphate molecule on the head. The actin is moved centrally toward the sarcomere, then the myosin detaches from the actin. The ATP molecule, which comes from the mitochondria, releases the myosin head from the actin and resumes back into a high energy state. ATP is broken down into ADP and P. The process of deactivation and reactivation continues, causing the myosin filaments to slide medially, crossing one another, thus concluding the muscle contraction.